Hi, today we are going to look at the complement system in the most simplified manner possible. The complement system is a part of the immune system consisting of a series of proteins found in blood plasma and tissues that work together to protect the body from infection. The complement system comprises of about 30 serum formation proteins grouped into complement proteins. The complement components are named by numerals. There are a total of 9 components from C1 to C9. And to remember, there are 3 subunits of C1 called C1Q, C1R, and C1S. The properdin system and the regulatory system, which we'll look ahead into the video, are named by literal symbols, example factor B. The term complement represents a group of protein normally found in inactive form, and when they are activated they augment the immune system and consist of about 5% of the normal serum protein, and the level do not increase following either infection or vaccination. So what do these complement system do? They provide resistance against bacteria, viruses, and fungi. So there are 3 main pathways called classical pathway, alternate pathway, and lecithin pathway. Let's look at them in a detailed manner ahead. Okay, so now let's take a closer look at the classical pathway. The classical pathway is an antibody dependent pathway. However, not all antibodies can bind to the complements of the classical pathway. So, to remember, there are major four steps that will come in all the 3 pathways, being initiation, formation of C3 convertase, formation of C5 convertase, and formation of the membrane attack complex. Now, in initiation step of classical pathway, the first step is binding of C1 to the antigen antibody complex. Okay, so out of C1, as mentioned, there are 3 subunits of C1 called C1Q, C1R, and C1S. Remember that C1Q is the first one to react with the FC portion of the IgM or IgG bound to the antigen. Now C1Q is an hexamer. As you can see that there are 6 heads, 1, 2, 3, 4, 5, 6. So it has 6 globular heads acting as a combination site. The effective activation of classical pathway will only begin when the C1Q is attached to the FC portion for at least 2 of the globular heads. As we can see, there are at least 2 globular heads attached to the IgG or IgM antibody. Okay, something you can remember here is that the IgG is monomeric, where IgM is pentameric. Okay, so since we know that IgM is pentameric and IgG is monomeric, so when there are 5 molecules, 5 sites where it can bind, so only one IgM can activate the classical complement system, while we need 2 IgG for classical complement activation. Hence, IgM becomes more potent for activation of the complement system. Now, all we have seen up till now is that into the antigen antibody complex, the C1Q complement protein attaches along with calcium. It activates the other 2 molecules of the C1, which are R and S. So when they are activated, note that C1S has an esterase property. So esterase, which means that it can cleave. Now this cleaves the C4 complement. The C4 complement protein is broken down into 2 further subdivided proteins called C4A and C4B. Now here you can remember is that C4B is slightly a larger molecule, and in other process you will see ahead, always remember that the bigger molecule will bind to the further activated complement cascade. Imagine this is a ball, a circle or a ball called C4, and it is broken down into 2. This is C4A and the slightly larger is C4B, and hence this is now going to be attached to the activated cascade of the protein. Now, C4A is an anaphylatoxin and its action we'll see ahead in the video. After C4 is cleaved and activated, now we have the antigen antibody complex C1 and 4B, which is the activated part. After the fourth is activated, now with magnesium and the C2 is broken down. So C2 is broken down into again two parts, 2A and 2B, but here 2A is slightly larger. Hence, we have attached 2A to this system. Now, we've already made this protein, which is C1, 4B, 2A. C1 comes from the attached C1 molecules, C4B comes from the C4 activated protein, and A molecule is now used in the other parts following the membrane attack complex. And the other is 2A. Now, this is called the C3 convertase because now this will cleave or break C3 into C3A and C3B. C3A is an anaphylatoxin which we'll again see in the membrane attack complex part, but C3B will now be attached to the already activated cascade of the proteins. What does this do? This will now activate C5 complement protein. C5 is again broken down into A and B. B being a little bigger will now be used in the other pathway, but C5B is slightly liable, which means it is not stable. And to stabilize, we need C6 and C7 proteins. So both these proteins now stabilize the C5B and makes C5B and 6-7. Okay, now we've made this, but there are two other proteins called C8 and C9. Remember that C7 and C8 have hydrophobic regions in them. So this helps in penetration of the bacterial, viral or the fungal molecule which we want to kill. Now the end molecule is called the membrane attack complex. Membrane attack complex is a combination of C5B, 6, 7, 8, 9, which we saw here. Okay, for now we saw that C6 and C7 was stabilizing the C5B molecule since it was liable. We also saw that C7 and C8 helped in penetration into the microbe or virus or fungus. And now the action of C9 is to form the channels or pore or perforations into the membrane. Imagine this is the wall of the microbe. Now what C9 does to it is that it will create pores or channels into the microbial surface. Now what pore does to it is that it will cause influx of water into the cell, which will cause cell swelling and hence thereafter cell bursting and lysis. Okay, so what happens after the membrane attack complex is formed? How does the targeted cell is lysed or damaged? Now, the first method of cell lysis or damage is by making pores or channels in the target cell membrane. Imagine this is a cell and there is fluid, extracellular fluid or any fluid outside the cell. Now there are pores or channels created so that this water moves into it and it swells up and causes bursting or cell lysis. This is the first method. The second being the inflammatory response. Now we had seen earlier that there are these fragmented proteins called C4A or C3A known as anaphylatoxins have been removed from this system, but it is now useful as all these anaphylatoxin they bind to the receptor on the mast cell and causes release of histamine and other mediators, which causes vasoconstriction and decreased cellular permeability. Okay, the other method is opsonization. Okay, what is opsonization? Opsonization is just making your food a little tastier. Imagine this is a bowl and it has food but it has no salt. What you do is that you add salt and spice to make the food tasty. Now, similarly, in bacteria or virus, to make the food tasty, you sprinkle or you apply the C3b and C4b. These are the opsonins, and on opsonization, there is increased chances or there is enhanced phagocytosis, so the cell will be engulfed and killed because it was phagocytosized. The third or the fourth is removal of the immune complexes from the blood. C4 C3b being an opsonin is also also recognized by the CR1. These are the receptors on the RBC. Now, what does this recognition by the receptor do? Is that it removes the immune complex from the blood and it takes it to the liver and spleen for phagocytosis. Imagine C3b is a car and the driver recognizes the immune complex and it takes to the liver where it kills it. The last one is viral neutralization. Now, this is very easy to understand that for any virus or a bacteria or anything to create any action has to bind to the receptors. Now, to the attachment sites has to be exposed so that it can bind to the cells and it can invade. But on what the membrane attack complex does is that it blocks the attachment site. Now, if the gates of a door, if a room are closed, how can you enter it? Similar way, if the attachment sites are being blocked, then there is no way that you can enter into the cell or the nuclear material can come out. This is the classical pathway. Similarly, we look at the Lecithin pathway and the alternate pathway in the next video. Thank you for watching. Like, share, subscribe.